Hey, and welcome back to Teach Me in 10, a video series brought to you by Technology Networks. Here we invite scientists to describe their research area or a topic in less than 10 minutes. My name is Rihanna Lilly, and I'm an editorial assistant for Technology Networks. Our guest here today is Dr. Martha Clokie, a professor of microbiology at the University of Leicester and the director of the UK's newly founded Centre for Phage Research. Today, Martha is going to teach us about the cutting-edge developments within the field of bacteriophage therapy, an innovative approach to combating bacterial infections. Okay, so over to you, Martha.
Thank you. My talk today is on the latest in bacteriophage research. So first of all, I have to tell you what bacteriophage is, and we can see in the top right of my presentation that here we have a bacteriophage. We can see they're a virus with a protein head and tail, and they generally have a double-stranded DNA genome. And all bacteria anywhere have these natural viruses, these natural bacteriophages that infect and kill them. And they're highly specific, and that specificity is dictated to by the tail fibers at the end of the tails. And what they do is they attach to the bacterial hosts and they become then viral factories. And actually, if we look at the other picture on my title slide, what we can see is lawns of bacteria, whereby there are holes in these lawns caused by bacteriophages. So one phage, exactly like this one here, has already attached. It's gone through many rounds of replication. Each time it kills a bacterial cell, it will release about 100 bacteriophages. These will then find a new host and so on. So by the time you see these holes, there's about maybe 10,000 actual bacteriophages in each little hole. So as predators of bacteria, they could be incredibly important in terms of really identifying novel antimicrobials. And the reason why this is so important is because we have a really impending crisis in terms of antimicrobial resistance. So this figure here is showing you the predicted numbers on the left of deaths that we'll get from bacteria resistant to antibiotics by 2050, unless we do something about it. So we can see it's predicted that up to 10 million people across the globe will die. And this isn't just an esoteric number. We can see on the other side, we can see the numbers of people that actually died in 2019. So if you look in yellow, we can see that if you include strict number of deaths from antimicrobial resistant infections and deaths that are attributed to the infections and another comorbidity, those number of deaths, really, they're more than a number of people that have died from dementia, HIV, AIDS, and malaria put together. So already we have significant numbers of people dying from infections. And this has really motivated a new look at bacteriophages as a potential solution. And what's really new in this field is that whereas 100 years ago, bacteriophages were first isolated, and our understanding of them was pretty blind, we can actually now very, very quickly go from isolating them to understanding what's in their genome and using this information to actually inform how we develop them. I'm going to show you some pictures of what we do in order, first of all, to bring them. But actually, first of all, I'd like to pose a question. How do you go from this, which is the estimated 10 to the 31 viruses on Earth? So we know there's more bacteriophages than any other biological entity. And this picture here is showing you seawater that's been stained with a cyber-green stain. And you can see all these big particles, they're bacteria, and all the little ones are bacteriophages. So they're really abundant, they're absolutely everywhere. But we want to use them as antimicrobials, so we need to find the ones that kill bacteria, particularly those bacteria that are resistant to antibiotics and those that cause us infection. So we want to know, how do we actually go from knowing that bacteriophages are everywhere to making actual pills or products that contain them. So the top of my slide, actually, here on the right side, we can see this is how bacteriophages are sold in some countries that use them. This is a Georgian preparation. So we can see they sell them in these little vials of mixed viruses that target certain bacteria of interest. But how do we do that within an NHS context? Well, what can really help us are the new insights that we can get particularly using genomics and also using artificial intelligence to help us interpret quite complex patterns. And together, these two really new tools will help us determine where phages are, which ones to use, what actual features in a phage are useful and are going to be useful therapeutically, and how we go about selecting those bacteriophages, and also which ones we should actually choose to put together in that final cocktail. So really, these genomics can really teach us about the where, the what, the how, and the who, and I'll briefly show that in my presentation today. I wanted to start by showing you that the where. So where do we find viruses? I've said they're the most abundant thing anywhere. Well, we can find them in association with soils, rivers, very often with sewage, where you have high numbers of bacterial concentrations in there, with also corresponding bacteriophages. And what you can see on this plate on the left, this is my somebody from my team holding at one of her plates. You can see individual little tiny plaques. So we bring them in from the wild, we isolate them. We then can take a single plaque. And previously, what would have been done is you could have propagated that plaque, made a lot of virus, and just seen what it killed. But actually now we can go from an individual plaque to a genome sequence in about 1 week. Perhaps it costs around £10, that kind of order. Whereas previously, when I first started working in this field 20 years ago, it took 6 months and cost tens of thousands of pounds. So you can see how accessible it is now. We can go from plaque to genome sequence. And that really allows us to be able to do all sorts of different things. So it can inform us, for example, how we actually grow the bacteria, and grow the bacteriophage interactions. It can teach us about the practicalities. It can teach us which ones we should choose, what should we prioritize, and actually, importantly, it can help us dictate therefore which ones we will choose for the production. So here on the right side at the bottom, we can see bacteriophages being produced at high volume that's going to be ultimately used in for a clinical purpose. In terms of knowing what those genomes encode, what we can do, we can take our genomes and we can show how related they are to each other. And this map is showing you many different bacterial pathogens. And if you look above, each individual dot is a bacteriophage genome. Now, if they're in a cluster, they're related to each other. If there's a gap, so within Salmonella, we can see the blue phages and the orange phages. There's absolutely no gene in common between these viruses. It's quite remarkable, the amount of genetic diversity. What we can do is we take the bacteriophages that we've isolated, we sequence the genomes, and then we can cluster them according to how related they are to each other. And then we can start to understand the biology of those particular viruses and identify, well, actually, is it always the blue viruses that seem to be aggressive? Do they work better therapeutically? And then we can go back to the genomes to work out why. So for the very first time, we can actually map the whole genetic space for the different types of viruses that target the different bacteria. And then we can understand which groups of viruses work the best. We can do this for all the different bacterial pathogens that exist. So even just by looking at these images, we can see that some bacteria have got quite different patterns of relationships of phages than others. So for the first time, we can zoom straight into the genomes and understand a lot of really important features. And we're just discovering all sorts of other amazing features of the genomes, like how methylated they are, and they're modified in different ways. And we can also really understand the relationships between what's in their genomes and their bacterial hosts, so we can understand that sort of whole arms race between the two. We can make sure that they don't contain anything bad in there, for example, any toxins, any antimicrobial resistance genes. So we can really, right from the outset, deliberately select bacteriophages that have got key genomic features, which we can then use to develop. So we spend a lot of time actually doing this. So it's my hypothesis really throughout this talk that phages will be able to transform modern medicine. They give us this whole extra set of tools. And how do we choose those ones? So I just showed you all those genome maps, and I said we can start to understand features. Well, what sort of features will we use? Well, one, for example, is going to be the receptors. So we can tell from looking at the genomes, we can start to identify the genes that encode for the receptors on the bacteriophages. So that's immediately, you can see key information. And we've written a whole lot of papers. I've listed them here, several papers that we've recently published. This is within my group at Leicester and various collaborators, particularly from Copenhagen. We can see one of the things that we have looked for are to choose viruses that, for example, don't integrate, they have the right life cycles, they don't have anything that's going to impede that successful infection. So although bacteriophages exist for all bacteria, the different groups are not equally effective. So by using these new tools and information from those genomes, that can really help us with the selection right from the very, very outset. So the last thing I really want to cover is in terms of how that genomic information that's new can help us get that translated product. So we had to take a step back here and really understand what is the information that's actually needed. Well, we clearly need to be able to produce bacteriophages under very, very high standards. We need to work with regulators to make sure that product, we understand what is needed for a product. And again, you can see that this information is really useful for a regulator because they want to absolutely be sure that your product isn't going to make a human worse in any way. So one of the things I did last year was I went to down to London. There was a UK government inquiry on bacteriophages in the House of Commons, and it was a really interesting inquiry. They examined all of the evidence globally from doctors and scientists, companies. They spoke with regulators from different places, and they came up with a really good report that essentially says that bacteriophages should be investigated. We need to have better integration between phage scientists and with regulators and people that can manufacture biological goods. And phages should be integrated into other guidelines. So by having that genomic information embedded within there, that can give the regulators the confidence that we're headed down a good trajectory to make a product that we understand. It will really help inform that as well. So in my brief overview today, hopefully I've been able to show you that bacteriophages, they're not new. They have this 100 years worth of isolation and characterization and history associated with trying to use them. But it's essentially, previously it was done blindly, but now we can actually delve into the genomics. This can really help us find phages for many new organisms. I think at the moment, something like 90% of all phages are isolated on 10 pathogens. We need to expand that, so that can help us with that aspect. So it can help us understand where they are. It can help us understand the novel content. When I talked about AI, there's many things like, for example, being able to move from protein space into structural space. We can then probe and be able to understand what a protein might be encoding, even though we don't know, we couldn't have recognized it before. We can actually recognize things a lot better. So we can, for therapy, the genomic information to that function. So we can find phages that have all different features that can grow at the temperatures and under the conditions that we use. I gave the example of receptors. We'll be able to understand genomes to really help us with the safety and the regulation aspects as well, and to be able to choose suitable cocktails that we can use to put together to minimize resistance. So we don't go down the whole problem of antibiotic resistance. We can not repeat the mistakes of the past. So I hope that was a helpful overview. I'm very can be happy to direct you to the further resources if you'd like to read more. Happy reading.
A big thank you for joining us here at Teach Me in 10. If you'd like to learn more about bacteriophage therapy or Dr. Martha Clokie's work, make sure you check out the resources linked in the description box below. Stay tuned for our next installment of Teach Me in 10. Bye!